HFR is a type of the F plus strain. Okay, so this basically is a derivative of the F plus strain of bacterium, or type of bacterial cell. The difference is it actually, instead of being a little standalone plasmid, it integrates itself into the chromosomal DNA. Okay, so what happens is you have your little F plasmid, and you also have the chromosomal DNA. And recombination events, this is somewhat rare, but again, we can recover it with bacteria because there's so many of them. But this will actually, you guys are not responsible for understanding the molecular mechanism, all the proteins and how the nucleic acids actually integrate. But this circle essentially gets cut, cleaved, so that it becomes a linear fragment. And then those two ends insert themselves into the chromosomal DNA. So that in this drawing, we kind of simplified with colors, but you basically have all your regular chromosomal genes that are found on that bacterial genome, and then you have your F plasmid that's in a continuous ring now with the original chromosome. So this is an F plus strain that has had the F plasmid integrate into the chromosomal DNA. So now this is referred to as an HFR strain. So we're kind of just, right now we're just walking through some basics so you guys understand when we talk about, if I'm asking you a question about an HFR, markers or genes, loci found on an HFR bacterium, you know that that means it's capable of being a donor. It acts like F plus because it has that F plus gene on it, right? It's just, now it's integrated into the whole genome. Okay. So as opposed to being a separate little plasmid, it's part of the full chromosome. Yeah.
The HFR stands for high frequency recombination.
Yeah.
Right. What is that exactly?
We're about to get to that. Yeah.
Okay.
Yeah. Good question. Yeah. So HFR actually stands for high frequency of recombination. They named it that because they noticed that there's a whole bunch of recombination that tends to happen with those strains. So what we're going to get to next is this concept of recombination in bacterium, which is a bit different than the way it looks with eukaryotes. So I'll give you an example. So we have an HFR cell. And he's, she, it, I guess, is getting friendly with F minus cell. I totally was telling myself this while I was writing this lecture. I hope you guys do the same thing. Try to make it exciting, you know, so look at this, HF4, hanging out with F minus. Alright. So there's, you can draw alleles and you can make up any allele symbols that you want to make yourself, to make this make sense in your head, right? So I think I just picked A, B, and C for right now to keep it simplistic. We're not putting the leucine and arginine and all those 3-letter codes. We're just going to think about gene markers and what might happen if it transfers this over to an F minus who has the opposite alleles. So maybe the F minus for A, B, and C, it has the recessive minus allele for those, the non-functional allele. So what happens, and in this chromosome you have your F plus gene, you have part of the F plasmid that's then integrated in. We keep using colors, but obviously this is just nucleic acids, not anything special other than the sequences themselves, right? So if this HFR strain, starts to transfer over, starts to unrolls and makes a copy of this chromosome, okay, and starts to transfer genes from donor to recipient. If it happens to pick up the locus, the loci for A, B, and C as well, so if it starts to send those in, what your resulting recipient cell is going to look like is you're going to have a fragment, right? You're going to have a fragment of DNA that matches, it's essentially homologous, different alleles, but you have like a halfway diploid scenario going on here, right? So this actually ends up being converted to double-stranded. It's still linear. It comes in as a single strand, single copy. The recipient cell will convert this to double-stranded. And sorry, I should be depicting this as the whole thing. You still have a helix, okay? Right? The bacterial chromosomes are always double-stranded helix. They're just arranged in a circle versus linear. So always in your little drawings, if it helps you to remember that, you can draw that double strand, however it is that you visualize that. I know I've been drawing it as a single line. Okay. So we have these same loci lined up here. What they tend to see happen with this HFR strain, which is getting at that question, is we tend to see recombination between alleles. Okay. So we tend to see recombinants formed. So these strands will actually, strands of nucleic acids will actually come in and invade each other, and recombination occurs just like we think of in eukaryotic chromosomes. Okay. But this is more of a random event, and the proteins that drive it and that cause it to actually happen, you guys don't need to worry about at this stage. But what we have is recombination between these genes such that you might end up with a recipient. The result of this, okay, would be a cell that has some of the genes, some of the alleles from the original strand, so maybe A minus, but B and C plus, say, depending on how the fragment, what point in the sequence that it actually recombined. Okay, and then you still have your other little linear copy stuck up here, which is now A plus, B minus, C minus. So all the properties, the kind of conceptual framework behind recombination is the same. So however you think that, consider that to be the same. The one main difference between bacteria and eukaryotes is we want to have a circular product after recombination. So can the strands cross over once? Or does it always have to be multiples of 2 to get a circular product? Totally set those questions up so I make it sound like there's definitely a right answer. Yeah, you can't. So one crossover event, try to visualize that in your head. If you have a ring and it splits and it crosses over once to the other linear fragment up here, all you're going to have is a big, long, linear fragment. Bacteria don't like that. Okay, that makes them very unhappy and actually will result in a non-viable cell. It has to invade and then come back down to complete that circle. You have to have at least 2, or 4, or 6, or 8. It has to be in multiples of 2. Okay. Otherwise, all you have is 2 linear products, and the bacterium needs to have a complete circular genome at the end of this recombination process. Okay, and what actually happens, and the reason that they aren't viable, this fragment here will be degraded. This will go away. Okay. After this recombination event occurs, that linear fragment will be degraded by enzymes and things found in the cell. Yeah.
But it can happen, like, you can have two recombinations between the B and Z? Or does it have to be between certain ones? Like,
In terms of where, like
Yeah.
the markers, it can be anywhere.
so it can be like 2 or 4 in between the B and C?
Yeah,
Okay.
yeah, depending on how far apart. And then again, the same
Yeah.
principles apply that we thought about with gene distance. So if two genes are really far apart on a bacterial chromosome, you could have 8 crossovers occur between them. But if they're like 10 nucleotides apart from each other, you just don't have the physical space to be crossing back and forth that many times. So the same concept that we talked about before with recombination applies here, and that the closer together two genes are, the least likely you'll have recombination occur between them. The other thing with recombination in bacteria, you always have to have sets of 2, and 4 recombinations are much more rare than 2. Okay, so again, you guys kind of have in your head from that 3-point cross that the double recombinant is the rare class, right? So a double recombination event is more rare than a single in eukaryotic systems. With bacteria, that same, just double it times 2, same concept applies. A 4 crossover event recombinant is more rare than a 2. Just because of that physical space it takes to make that happen.
Why is this one lost again?
It's bacteria have evolved to break up linear fragments of DNA in their cells. They usually treat it as viral DNA. It's an evolutionary mechanism they've evolved. Same often in our cells too. Little fragments of DNA get degraded over time. Enzymes and things that are in the cell. Things like restriction enzymes. We talked about restriction enzymes. Those are originally derived from bacterial cells that break up stuff like this because they've evolved to consider it to be viral DNA. They're trying to guard against virus DNA coming into their cells.
There was a recombination between A and B and another one between B and C. Would it be A minus B plus C minus?
Yes, if you did it outside
B.
of B, yeah, the way I lined it up.
Okay.
So like, the product I drew over there on the left, I should have made this look more like recombination here,
Okay.
right?
Yeah.
Yeah, so recombination, getting at your question, can happen wherever you'd like. Well, not wherever you'd like, but whatever, where it has multiple possibilities. And so you can end up with different allele combinations. If we recombine totally on the outsides of this fragment, you could completely exchange your alleles for donor and recipient, such that now you have A plus, for all of the alleles and A minus on the fragment that gets lost. A, B, C minus on the fragment that gets lost. Yeah. So same kind of ideas about recombination occur. And in the HFR DNFAS question, it just happens really frequently. It's a property of having that as a plasmid integrated in to the genome. Okay. Make sense? Everyone's kind of sleepy time still. Okay.
So there's no like percent or anything for HFR?
They're percent recombination,
Yeah.
or yeah, and we're gonna get to that. There's
Okay.
definitely
Okay.
you can definitely look at how often the recombination events occur, and the same concept of like a 4 event recombine is smaller percent than the double
Okay.
recombinant. Yeah. So we can do things. That's a question. We can do genetic mapping using this concept that's happening here. Okay. And these kind of studies are actually, it's really exciting. You guys all wake up. You get to interrupt bacteria having sex. This is what you get to do. Okay. No, nobody likes that. Let them finish.
Go for a walk.
Go for a walk. I do not miss having Okay. Alright. So, there's this concept called interrupted mating conjugation experiment. So you let two bacterial cells come together, form a pilus, start to exchange genetic information. Okay, and I'll draw out this process first. I'm going to say it out loud, and then we'll kind of draw a diagram. So you let them start the process, and you can interrupt it at different points in time, because this takes time for the DNA to travel across the pilus and move from one cell to another. So you can see different genes coming in. Everyone's like laughing, so they're thinking about it. You can see different genes coming in. The longer you wait, the more genetic material makes its way over into the recipient cell. Okay? And they actually literally do this by, you can use a blender, or you can shake the flask. I've seen people actually do it in lab. You can shake the flask really vigorously, and it breaks that pilus, and it's kind of sad. And they disrupt, okay? So whatever got over, whatever this DNA material that made its way over, that's all you have, and it gets cut off from there. Okay, so that's kind of the concept behind this. So you can start with 2 cells, all right? Your HFR and your F minus. And I'll just keep doing this color different so that it's. So HFR is going to unravel its chromosome and make a replicate copy of it, a linear copy that it's going to send over. And if we have all these genes along the length, so I'm kind of just numbering them 1 through 7. If you stopped it and interrupted this experiment here, maybe 10 minutes, bacteria take a long time, 10 minutes after this conjugation event has begun, you might not even have any genes that have made their way over if you disrupt this too quickly. Whereas so I'm kind of doing a time lapse drawing. You guys act like you don't like the analogy, but you totally do. Or bacteria. Okay. Whereas if we waited another, 7 minutes, maybe you would start to see if this pills broke here, you might see genes 1, 2, and 3 have made their way into the recipient F minus cell. And the concept you can keep kind of going where now if you waited, 25 minutes, we would have the whole thing in. Or a longer fragment.
Does the donor's genetic material copy as it's transferred, or
Yeah.
completely before?
The donor actually is making the copy as it's unraveling and transferring. Yeah, so it's kind of happening in real time, so simultaneously as it's being transferred. Yeah. Okay, so here's maybe 25 minutes. So what researchers have actually done, and then the other key thing is this F factor that creates it to be from an F plus to an F minus, or an F minus turns into an F plus, is always last. So whenever you're looking at a set of genes, the last thing to enter the recipient cell is going to be that F factor. And then what about the early, so I happen to number these in like sequential order from the right hand side. We started with, oh, let's do that. We started with like gene 1, 2, 3, 4, whatever. But what does that tell you about those early markers? Are you more likely to get those transferred or less likely? More likely, yeah. The earlier the markers are, the farther away they are actually from the F factor, more likely you'll see them transferred. So that's where also your percent numbers, we were talking about recombination, But in an interrupted mating conjugation experiment, you're going to see high numbers of cells that have early markers transferred. It's much more likely to get those first few genes in than the later few. You'll have fewer cells that make it through this 25-minute experiment, or make it to getting the entire fragment in, especially if you're shaking that flask all the time. Okay. So this is what people do. They actually allow conjugation to occur and then disrupt it at different time points. And then you can assay the bacterium by growing them on different plates, right? So conceptually what's happening in the lab, if you stopped here, and we only have genes 1 and 2 in, and maybe those are genes that confer resistance to an antibiotic. Okay, maybe gene 1 is a strep-resistant gene. You could then plate that bacterium on streptomycin and check to see who received that first gene. And so this is how they actually, we're depicting this out because they know this is what occurs, but how they test it in the lab is then you plate them on different types of medium, you put different antibiotics, different nutrients, and you can test what each marker is coming in, okay, or whether it's made it in or not. All right. I'll give you guys some numbers so we can kind of think about some examples of how this might work. But just conceptually, the interrupted mating experiment is clicking for you guys. Okay. Wait for 5 seconds because you're thinking about bacteria sex, and then back to sleep. So here's an example. You take an HFR. Take your HFR strain and your F minus.
And do we know what those are already?
Hmm?
Do we know what genotypes those are already?
So what I'll do, I'll present this, so in black, I'll write up here the information you would be given if this was a problem, okay, and then we'll kind of walk through. So this is what you would see on your page, is HFR by an F minus cross. You'll be told that another piece of information you would be given in this type of problem is a gene that enters first, so Lu plus enters first. So you're already given that first gene. What this problem is getting at, though, is the order of the other markers is unknown. You're also told, so getting your question about genotypes, you're told that HFR, in terms of genotypes, so HFR is fully wild type for these markers that we're looking at. So normal. And in this example, F minus is the auxotrope. So then what's done is Lu plus recombinants are selected. So Lu plus recombinant. And you end up with the following numbers. These are all in percentages. So percent of your total bacterial population of cells. So you have 27% isoleucine, 13% Armel, 82% threonine, and 1%. So what the question, so this would be all the information for this particular problem that you'd be given. Okay, you're told You made a cross between HFR and F minus. Lu is the first gene. Order of the other markers we don't know. So that's essentially what the question is asking you, what the order of all of these genes are on this fragment of DNA that's coming into the recipient. You know that HFR is all wild type and the recipient is all an auxotrope. So you're looking for places where it's recombined and how frequent that recombination occurs. Okay, so we know the first gene. The very first gene to come in is leucine, because that information was given. When we thought about this, we thought about the early markers are much more likely to come in, right? So you're going to see a high percent of recombinants recovered that have early markers that are close to leucine will show up on our results as a high percentage, right? So what's our next one? Yeah. 82%. That's being transferred much more likely during the conjugation event, along with leucine. That's a high likelihood of them both being transferred together. And then leucine recovering the recombination that gives you plus prototrope phenotype. Because your original strain, your original recipient is auxotrope for all of these genes. What's our next gene on the board? Or I guess going from IL, and then at 27%, and then 13% we have MAL, and 1%, our very last gene over here is TRIP. So for this particular type of problem, I'm not asking for distances or map units between them. Often with the bacterial stuff, you're just giving the order. And we're going to talk about, I'll try to put together an example for later in the week, or at least before you guys have an exam. And I think you might have a homework problem that actually uses, again, this is before anybody knew anything about nucleotides and base pairs. So just like we talked about recombination units being map units or centimorgans, those are kind of this weird fluffy unit, right? It's like an arbitrary unit we're just applying to say it's a relative distance. In bacterial genetics, they actually use minutes as one of their units, okay? So they're timing the minutes it takes between entry, and so I can walk through an example of what that looks like. They know at 10 minutes, threonine came in. 10 minutes after leucine, we see threonine. 2 minutes after that, we see isoleucine. 3 minutes we see MAL or whatever. So they actually, you can do units between here, and there are different varieties of them. One is minutes. There's also a way to use this percent recombination. So we can walk through examples of that as well. But essentially, you can get this order knowing the logic that we just walked through. Okay, I wanted to, before we got into doing units and stuff, I wanted to do another example of multiple crosses. So any questions about, I just raised it, this problem that we just did? Up to that point, not putting units on or anything in it. Okay. So we have E. coli. In this case now we actually have 4 strains as opposed to just the 2 that are making. Okay, so you're going to be given a table. So you have 4 HFR strains, and you know that they're donating a bunch of genetic markers. And the reason I use the term genetic markers and not genes is some of these might not be actual translated product type of genes, but they're markers that we can track on the DNA. Okay, and you'll have this a little chart that tells you strain 1, 2, 3, 4. And the markers that they donate, and they're shown in the order that they're donated in.